economist at Harvard College. And at a New Year's Eve party, he met an unknown biologist who invited him to sail across the Atlantic. He said yes. Came back. That biologist is Craig Venter. Juan came back, started life sciences project at Harvard Business School, and now is a venture capitalist in Boston. And here he is to talk about our future. Thank you.
All right. So I know what all of you are thinking. There's nothing quite like a little bit of late afternoon genetics to get the blood rolling. And it's a great thing for a siesta. And as you're thinking about this stuff, and you're thinking about what the economy looks like these days, it's a little bit like swimming in Maine, right? And it ain't terribly pleasant. But the real issue here, and what's really interesting, is even as we're going through this horrible financial crisis, and even after country has this huge debt issue, there's a second and even bigger wave that's taking place, and it's going to take place regardless of what happens in Afghanistan or what happens in Iraq or what happens in the economy and the stock market. And the reason why is because you have this huge wave of technology that keeps washing over societies at different times. And that wave of technology keeps moving across, and it's going to keep moving across, but it will not do so in an equal way in all places. And when you think about what's driving that technology and what's going to drive this talk, because it's an afternoon talk, I want to make it very simple and just come up with about 3 very simple, silly ideas. Here's the first idea, right? So almost all wealth derives from code. The second idea is code keeps evolving. And the third idea is we're moving from digital code to reading life code, to copying life code, to actually writing life code. And that's the entire talk. Now you can have a nice siesta. And as you're thinking about how this stuff is structured and why it's so important, the basic thesis is all wealth derives from code. And the reason for that is because we have a tremendous overlap in genes with a whole bunch of animals. The difference between men and mice is about 5% of gene code. Almost every wife in the audience already knows this. By the time you get to monkeys, the difference is about 1.23%. And maybe dolphins sing, and maybe whales have songs, and maybe chimps have a sign language, and maybe dogs can learn commands. But there is no animal on Earth that systematically teaches stuff to its great-grandchildren across time. And that's why code is so important. Because when we start doing something that looks like this, what we're doing is we're actually telling people, look, this is how you have a baby. This is the kind of fish we eat. This is the music that we play. This is how we dress. This is how many of us there are. And we have actually just learned an awful lot about Argentina 2,000 years ago because we code. Now that's enough to generate a tribe. An empire looks like this. And the reason why this is an empire is 2 very important things have happened. One is when you can move code, when you don't have to go to the cave wall to learn, when you put it on papyrus, when you put it on clay tablets, then you can spread code over a vast distance. The second thing is when you standardize code, everybody in Upper Egypt and Lower Egypt can read exactly the same thing. Then you can have a holy book. Then you can have a customs union. Then you can have textbooks. Then you can have people learning the same stuff across time. And that's why code is so important. This particular sentence, written in Egyptian hieroglyphs, is perfectly innocent. It simply says, Red Sox rule. Well, the Egyptians were smart. Then you go into a 20-some-odd letter alphabet, and then you put it in a movable type, and all of a sudden you have huge libraries and huge depositories and great bookstores. And you walk into the bookstore here, and there's a lot of code in that bookstore. There's a lot of knowledge. There's a lot of accumulated thoughts that you read and then you pass on to your grandkids. Now, what happened over the last 30 years is we started to change that code again, and we started to write in these little ones and zeros. And what we did is we collapsed every word written and spoken in English into 2 letters. This particular sentence here, if I send it to you on a BlackBerry, the first line will read, I love you. If I'm in a bad mood, I send you the second line that says, I hate you. So the function fundamentally changes depending on whether you get green or purple. And what you've done is you've collapsed every word written and spoken in English, and French, and Aramaic, and Egyptian, and Japanese, and Chinese into a 2-letter alphabet. And you've also collapsed every bit of music and every tonal scale. And you've collapsed every photograph, and every video, and every film. And when you do that, you have this incredibly powerful language which allows you to take different alphabets, different notation systems, and it's so darn powerful that this is the main language in the world today. Right? About 99% of the data transmitted in the world today is transmitted in this language. So if this stuff is so powerful, why weren't people using it? Well, among other things, because this was 5 megabytes in 1956. Pull out your cell phone, take one photograph. You've just used up $1 million worth of computer memory. You probably don't want to take a second photograph until the stuff starts halving in price and doubling in power. And when that happens time and again and again and again, we're going to double the amount of photographs taken by the human species over the next 3 or 4 years. Because digital cameras, it's irrelevant. You don't have to go and chemically print, and you don't have to pay Kodak, and you don't have to go and pick it up at the drugstore, et cetera, et cetera. It becomes a much more powerful system. Here's a different way of looking at this system. This was the world's most powerful computer in about 1996. This is the first time a computer can make 1 trillion floating point operations per second, so 1 trillion calculations per second. Really powerful stuff. About 3 months ago, I introduced the Intel chip. This is the newest version of an Intel chip. It's about the size of, a coin, a dollar coin. And this little thing has about 2.3 billion transistors in it. These things network together. You can plug them in like Legos. You plug in 2 chips about the size of a dollar coin, and you have the equivalent of 1 trillion floating point operations per second. So you now hold inside your laptop the world's most powerful computer in 1996. And oh, by the way, these little things network, and you can plug in 256 of these things together, so you can have a single blade with 128 terabytes worth of power in a computer. Let's put that in context. That's 5 libraries of Congress. Right? That's why this code is so important, and that's why understanding code is so important, and that's why code generates wealth. Now what's really important, what I really want you to walk out here with, is the code is changing again. So in the same way as most of the wealth generated over the last 30 years, in Malaysia, Singapore, Thailand, Bangalore, Silicon Valley, Denver, Austin, etc., has been based on the ability to manipulate ones and zeros, to make Hewlett-Packard, to make consulting, to change media, to make digital photography, to make YouTube, to make Google, to make Skype, etc. Now what's beginning to happen is we're beginning to use a code that was created. Mendel started studying this, and Darwin studied this. But Watson and Crick in '53 started arguing that all life on this planet is coded in 4 letters, 4 letters of DNA. They called it a double helix, so it's a little spiral staircase. And it's got 4 rungs on this thing. It's got adenine, threonine, guanine, cytosine, all life, 4 letters. Every clover, every orange, sheep, cow, human being, politician, they're all made of the same stuff. And if you stick that into these machines, you can begin to write life code, right? Because these gene sequencers will tell you what the gene sequence is of a particular life form. And you can write unbelievably boring books. Right? There are single machines today that can sequence human genomes on a little desktop. And they're producing about 100 billion letters of gene code per day. These are the world's largest databases. These are accumulating 37% faster than computers can be built to keep up with this. If you're working at Google, if you're working at Microsoft, if you're working at Intel, these are interesting problems to have. If you're working at EMC, these are interesting problems to have. This is going to use up huge amounts of bandwidth. Let me give you an order of magnitude. The human species generated the first zettabyte worth of digital data in everything we do about 3 years ago. A zettabyte, as all of you undoubtedly know, is 10 with 21 zeros. Single hospital systems will be using and producing a zettabyte worth of data within about the next 5 to 7 years. Right? These are absolutely massive data explosions. Why are these letters so important? Because these letters are how life is coded. So if you take this little apple, this is a program to execute a life code called Apple, the real one. And the way it works is you don't plug it in, you do this. And it begins to execute a code that looks a lot like that. ATACAGG make a little root. TCGAA make a little stem. AGCAA make a series of flowers, make them white, and then make a couple of copies of this thing. Remember those little ones and zeros? 0, 1, 1, 1, 1, 1, I love you. 0, 1, 1, I hate you. The function fundamentally changes depending on the order of the ones and zeros. If I change a couple of ATCGs in this thing begins to look like this. And if I change a few more lines of code, then an orange becomes an apple. Or it becomes a tangerine, or it becomes a grapefruit. If you change a few letters in your gene code, and we're not talking about a lot of gene code, then you become the person sitting right next to you today. Be a little more careful where you sit next time. And as you're thinking of what it means to be able to code life and what it means to be able to structure this, it means that all life turns out to be imperfectly transmitted code. And we're beginning to understand life as code, and we're beginning to read that life code. And as that goes forward, that's the reason why you get evolution. Which, by the way, is not a theory. There's some pretty definitive proof. We're just not quite sure which direction it's currently taking. These dogs have about 23,000 genes. You change one gene out of 23,000, you get a 200-fold differential in body weight. It really matters being able to read and write this life code. If you're in the business of agriculture, if you're in the business of growing stuff, if you want to make biofuels, you really better understand that a gene will lead to a 200-fold differential in output. And as we begin to read this stuff, it's spreading very widely. So this is a gene cloning lab in Argentina. These are very nice people, and as you talk to them, this little guy shows up. This guy's been treated so nicely since it was a little tiny calf that it acts like a Labrador retriever. So it comes up, it waits to be petted, you're petting this guy, and then these two guys show up. And at that point you say to yourself, hey, you know, self, those two look really similar. And in fact, they look an awful lot like that one. And the reason why is because each of your cells contains your entire gene code. Each of a cow's cells contains its entire gene code, which means that you can take some cells from the right ear of this cow, insert that gene code into a fertilized cow's egg, and give birth to 2 clones. So this is the second part of the story, right? The first part is you can read life code. The second part is you can copy life code. This is gene cloning. This is a vet who's got his left hand way up the back of the cow. He's holding an ultrasound in his right hand, and he placed several dozen cloned embryos in the morning that I was there. Which is why when you walk out on the Argentine Pampa today, it turns out there's actually an awful lot of cows that look really, really, really similar. Because I did not expect to find this in Argentina, I'm running around taking pictures like a madman with my digital camera, until a very friendly Argentine cowboy, a gaucho, shows up and says, "But Che, what are you doing? Why are you taking so many pictures? Can't you understand they're all the same?" Here it begins to get more interesting. This animal was born a few weeks after I took those pictures, and this animal is particularly important because they changed a couple lines of gene code so that this animal would produce a medicine used to treat cancer in its milk. So it's a copy, but it's a modified copy that also makes a medicine. It makes erythropoietin, EPO. 20 of these animals substitute for a factory that's about that size. This is Genzyme next to the Harvard Business School. how we make things, where we make things, is going to change on a scale similar to that of the Industrial Revolution that's already happening. Because we're beginning to understand how life is coded and how life makes things. How it makes textiles, how it makes medicines, how it makes energy, how it makes a whole series of things. Of course, Americans are different, right? So we would never accept ever having a medicine in our bodies made in a cloned cow's milk, which is why a subsidiary of this particular company is using goats. Now, there are 150 goats in western Massachusetts that are making an EPA and FDA approved medicine that is now being used for the likes of all of you. This stuff is moving very quickly. Now, when you begin to see life as code, then it's not just the body that's code, it's each of the cells that is code. What's interesting about that is you don't need the animal to make the code, right? That means that you can make cloned mice teeth that grow without the mice. And you can make them in petri dishes. And of course, if you can make cloned mice teeth in petri dishes, you can make cloned human teeth in petri dishes. This should not surprise you. You are born with no teeth. You grow a full set of teeth. You give them to the tooth fairy. You grow a second full set of teeth. But if you lose that second set of teeth, they don't regrow unless if you become a lawyer. Most of us only get 2 shots. But of course, because we know how to make teeth, because we've already done it once and twice, your body knows how to make those teeth. So it's reasonably simple to take a biodegradable mold of a tooth you lost, take some adult stem cells from one of your teeth, put it on that biodegradable mold, and grow yourself a third tooth. Your body knows how to do that. If you can do that with teeth, you can also do it with ears. You've already made 2 of them. If you can do that with ears, then you can make it with bladders. This is work by Tony Atala down at Wake Forest. He's an incredibly nice man. You meet him in the street, you think he's, a modern saint. And he appears so kind and so self-effacing. But Tony's now made bladders for a whole series of women who've lost their bladders to cancer. And it's a whole lot better to remake your own bladder on a biodegradable mold than it is to walk around with a plastic bag the rest of your life. And oh, by the way, as you go into Tony's lab, he did the first transplant in 2006, and he's now working on about 42 different organs. Your kids are likely to live 20 or 30 years beyond where you think they're going to live, and they're likely to be running marathons at age 100 on regrown body parts on whatever beach or Aspen they want. Right? And this is going to change things in a very substantial way. When I was at this lab about 2 months ago, I picked up these little inkjet cartridges. And Tony turns around and says, please, just put them down for a second. And I did, and then he explained to me that these inkjet cartridges, like you use for your printers, were full of cells, not full of ink. That he was using them to print layers of skin. Now, that may sound wildly exotic, but of course, last month I got the Harvard Biosciences catalog in the mail. And if you'd like to try this at home, this is one of the pages. And you can buy your own 3D bioreactor to make hollow organs or bronchia or trachea or blood vessels if you'd like to try this at home with your kids. Or maybe you want to try a larger science project. You can print mouse hearts that beat. This is not science fiction. These are pictures. This is historical stuff. I'm not a futurist. I use photographs because I want you to understand what's already happened, not what's going to happen. And as you start to go through this stuff, then it begins to get a little bit weird. This is an experiment done about 8 or 9 months ago. And what these folks did is, remember, each of your cells has all of your gene code in it. So they took a skin cell from a mouse. They de-differentiated that cell into a stem cell. How does that work? When your mother and father come together, they create one cell. Right? That is a pluripotent stem cell. It can become any part of your body, and it does. That one cell becomes 2 cells, becomes 4 cells, becomes 8 cells, becomes 16 cells, becomes 32 cells for about the first 16 divisions. Those cells can become any part of your body. They are pluripotent stem cells. After 16 divisions, what starts to happen is those cells keep the entire gene code, but they decide to become specific parts of your body. I'm going to differentiate to become bone, I'm going to become stomach, I'm going to become arm, I'm going to become leg, I'm going to become eye. But each of your cells has your entire gene code. So think of it as going up here to Ajax. You're sitting at the top, and you have the potential to go down any slope on that mountain, and there will be different consequences depending on which slope you take. Now, the interesting part of it is when you invent a ski lift. And that's what these guys did, right? They invented a way, first, of taking a skin cell and making it into a stem cell. And then they found a way of making the stem cell become the animal again. So what happened with this is this particular skin cell gave birth to a full mouse. Think about that for a second. You take a mouse skin cell, you turn it into a mouse stem cell, and that stem cell gives birth to an entire mouse. Men are now officially useless. We knew that, right? But anyway, as you're looking at this stuff, one of the really interesting bridges that's occurring is this bridge from academia into business. There are societies that adopt and adapt, and there are societies that don't. When a society doesn't adopt and adapt, that country, or that country's gods, or that country's industries tend to disappear. We no longer, most of us, pray to the god of rain, or the god of maize, or the god of the sun. There's a whole bunch of industries we're no longer in. And 3/4 of the flags that are at the UN today did not exist 60 some odd years ago. What makes countries rise and fall? What disappears industries and creates industries? It's this ability to adopt and adapt, to quote Darwin. There is a really powerful trend in the United States that's able to take translational research and turn it into businesses. And let me contrast 2 societies. I don't think there's a smarter group per capita on the planet than the Brits in terms of life sciences. When you think of their history, these are the folks who created antibiotics, they created monoclonal antibodies, they discovered DNA, they did cloning. But there is an ethos inside a lot of those universities that you should not have rich academics, much less rich graduate students. So you drive outside of some of the molecular biology departments, and there's a whole pile of bicycles there. When you contrast that at Stanford, where there's a pile of Ferraris outside, or MIT, there is a very different ethos in terms of we are doing this strictly for the good of mankind in research, or we're doing this for the good of mankind, and we can translate into real businesses. And what I want you to understand is all of this molecular biology stuff that's been going on since the 50s is now reaching prime time and is beginning to change very, very large businesses. We are now able to program things like bacteria. To pick this example right here, we can now program bacteria in such a way that it can make a biodegradable plastic. About 93% of the weight of this bacteria, metabolomics, is a biodegradable plastic. But it's not just little Cambridge startups that are using this stuff. It's little startups like DuPont. What happened at DuPont is they got tired of depending on a whole bunch of oil-producing states, which were yo-yoing the price of petrochemicals. So they sold off the nylon, polyester, rayon, all the stuff that was made out of oil and petrochemical derivatives. And they started making their new things that make the stretchable, wearable fabrics, that make your tennis shoes, that make the new stain-proof rugs, in a factory in Tennessee inside bacteria. So this looks like a giant beer factory, except that what you're making is fabrics and textiles and chemicals. And oh, by the way, This is now 33% of DuPont's earnings. These are not small changes. These are not just small startups. This is General Electric. About 14% of General Electric's income is now coming from healthcare and life sciences, and it's the fastest area of growth. This is something that's beginning to work its way across IBM. When you look at where the massive data projects are, how you fill the internet, how you use data at Google, at Microsoft, at IBM, one of the really interesting questions is how do you keep up with a system that you can't build enough computers to store the data coming out of the system by 37% per year? Here's a little experiment we ran in a company that I co-founded with the guy who sequenced the human genome and the guy who won the Nobel for restriction enzymes and a very smart IP lawyer. And what we did is we were sitting in an Italian bar in Alexandria, Virginia, and I don't think it had a lot to do with about the third scotch, but we started thinking, what would happen if we could program cells much as we program computers? What would happen if we could build a full program for a cell? And what would happen if we could get that cell to execute the full program? Well, this was announced about a month ago. And what we did is we built the world's first synthetic cell. How do we do this? Well, we took gene code out of a computer and said, this is the code we want to build. And by the way, we inserted 4 pieces of poetry into this code, just so people knew that this was our species. And then we had robots assemble these things into what looks like a March Madness bracket, or for those of you who are highly intelligent and evolved creatures, it looks like the World Cup bracket. And we assembled pieces of 1,000, and then we took 10,000, and then we took 100,000, and to make a long story short, we built the world's largest organic molecule. Nobody had ever come close to building a molecule on this scale. We then found a series of technologies, or a series of discoveries, for inserting that gene code into a cell and having the cell execute that code. And to make a long story short, we built the world's first synthetic cell. Now, there's a couple reasons why this thing is interesting. This is a software that builds its own hardware. And it's different from biotech because biotech is inserting a single gene into a very large set of life programs, hoping that that single gene change will make that cell do something useful. Let me put that in context. Take a single sentence or a paragraph, insert it into a Tolstoy book, and hope it changes part of the outcome substantially. Exactly the right sentence in exactly the right place might do that, but you're going to run through a whole lot of experiments where you're going to get no change. But if you can program this thing from scratch, and every sentence and every page and every paragraph of the entire book is written from scratch, then you have a capability that looks to me like the first transistor. And in the same way as we built a huge IT industry out of the Fairchild and then the Intels and the AMDs, I think this programmable cell is going to be what scientists in their highly technical terms call a BFD. That is one of the reasons why The Economist made this cover with this discovery, talking about the consequences of the first artificial organism and where it leads. And the answer is, honestly, we don't know. But I can tell you this is likely the birth of a very large new industry. And the US is a leader in this industry, and if we're thinking about where the next Intels and Googles and stuff come from, they come out of things that look like that. One of the first applications is we found a neat little startup in Texas called ExxonMobil. And we reached an agreement for them to use our cells, our programmable cells, to start making fuel out of algae. And we've reached an agreement with a series of other companies that want us to program cells for their specific purposes. They are not small companies. I think the last thing that I'm going to say before we go to questions and answers is I really want you to understand where wealth comes from. Wealth doesn't come from imperial buildings in DC. Wealth comes from 2 or 3 story buildings that looks like that. Here's what the numbers look like. About 0.2% of US GDP has been invested in venture capital. The companies that have been venture backed are about 11% of US jobs and about 21% of economic output. You want to understand which regions, which countries are going to be rich and which are going to be poor, go walk around the universities. Tell me who's got really great research, and then count the number of startups around that university. Because that's what builds the Silicon Valleys, that's what builds the Taiwans, that's what builds the Koreas. You get that part right, you get your Bangalores. You don't understand this stuff, you don't understand this translation between code and how code changes, and the economy and new businesses. And your country and your industries are likely to at least become poor, if not disappear. These are very, very large transitions that take place. Imagine the US without any type of an IT or digital world and what this economy would look like. So what I want you to do is not be scared of this stuff. Right? Don't walk out of this thing looking for the nearest open window and jump. I want you to walk out of this thing as excited as you've ever been for yourselves and your kids. Because we just touched in this hour a very, very small portion of some of the changes that are happening that people like Drew Endy are driving. Very small portion of what's happening in nanotech, very small portion of what's happening in robotics, very small portion of what's happening in computer research, a very small portion of what's happening in genomics, proteomics, metabolomics. We now have a giant smorgasbord of ways of building companies and industries that are very, very large. We now have instruments to build wealth on a scale which no other human civilization has ever had. And we've brought about half the world out of poverty using the previous code. Now we've got an even more powerful code. That gives your kids the opportunity to live longer, gives you chances to completely change businesses, gives you chances to build entire new economies, but we have to make our kids literate in this stuff. We've got to keep making science interesting. I don't understand how in the hell we can take science and this kind of an adventure and make it boring. Because for me, every bloody day as a venture capitalist is like Christmas. Because the smartest people in the world come through and say, look, I've got this vision for something I want to build. Help me build it. And then you read this stuff, and then you get the science articles and the nature articles. And so many people react to this stuff by being scared instead of by being incredibly excited about what we have. We're going to double the amount of data generated by humans in the next 5 years. What do you want to do with that? I mean, it's a hell of an adventure. So let me stop there and see if anybody has any questions, accusations, harangues. You don't have to put a question mark on the end of what you want to say. Please. Sir. Just try this one.
Thank you, sir. When do you think there's a realistic chance that a person like me who's practically blind could get a new eye?
You know, the questions that we're facing are just so fundamentally interesting and complicated because one of the questions is how quickly can we go after macular degeneration? Can we start making parts of the eye? Can we start making a series of operations? And there's a whole pile of really smart people working on that. And there's a real disconnect between how quickly we can discover stuff and how quickly we can improve it. And we have a huge problem in regulatory structures and how they're organized because we're measuring how many people get hurt by a medicine that comes to market. We're not measuring how many people we're hurting by taking 14 years to come to market with a medicine. And those are two important questions. We've got to measure both of those. Not to be flip about it, but the more interesting question is not just what are we going to do with current eye tissue, it's what are we going to do with eyes. And let me give you one example. This is what's happening with ears, okay? I mean, if you remember your grandparents, Very often, when they started losing their hearing, they got these giant cones and they'd try to hear through a cone. And then they got these giant boxes that would squawk at odd times during dinner. And now they're using these little things and you can't tell that they're wearing them, right? And these things have now gotten to the point where you can hear about 76, 78% discrimination in a room that's loud, which is pretty damn good. But now what we're beginning to be able to do is we're beginning to implant this stuff in cochlear implants in such a way that people who are deaf can begin to hear. And now people who are deaf can begin to hear at about a 70% range inside these things, and it's doubling in power and having in price every 18 months. When you have a system like that, it's going to get to the point where those who are born deaf are going to be able to hear as well as we can. And about 18 months after that, they may be able to hear better. They may be able to hear in tones that bats or dogs or whales talk in. They may be able to focus. They may be able to tune their hearing. And we may get to the point sometime in the next 10 or 15 years where you will not be hired by a top-flight symphony orchestra unless if you've had a cochlear implant. Eyes are about 10 years behind that. Some of the first eye experiments have been taking place in Germany over the last 3 or 4 years. And they're beginning to generate these little tiny eyepieces that tie into the brain. And they're getting to the point where you can discriminate between shadow and light. But in 2 or 3 years, then you're going to be able to discriminate shapes. And 5 or 8 years after that, then you may be able to see very rough things. And in 10 or 20 years, it may be that some people will have eyes that will allow them to see in ultraviolet or infrared or will be able to zoom. We're entering a period that's going to be a very strange period. And it's going to get really interesting. Shoot. Why don't we do this, gentleman, then we'll do the one in the back.
We're talking about the biology and the science. How about the sociology and preventing people from cloning evil?
You know, your question makes me think of a whole parade of people that I really don't want to see a whole 100 copies of. But I'm going to be sensible. I'm going to keep my mouth shut about who they are. Look, human beings, I don't think human beings have ever come up with a technology that hasn't been used to hurt somebody. We are spectacularly good at hurting each other and using technology to hurt each other, but I think we're also very bad at assessing risk. So here's what risk looks like to me. We have done absolutely horrendous stuff with steel, with computers, with electricity, with automobiles. One of the most dangerous things we do is to walk up and down staircases. That's one of the things that is really dangerous. But we have this thing where the new, the novel, tend to scare us a whole hell of a lot, and one example is genetically modified foods. There's a lot of people, especially in Europe, who are just terrified of stuff. And I was in Paris 4 days ago, having a debate with a nice French scientist who was telling me how outrageous it is that we're putting genes in our food. He's doing this with a cigarette hanging out of his mouth. and what you really have to look at is food is really dangerous. For most of human history, one of the most dangerous things you could do was to have a drink of water. That's why so many civilizations made wine or beer part of their rites, because it was safer if you were part of a rite that drank wine or beer than if you drank fresh water. A lot of foods, peanut butter, gluten, honey, you name it, have led to violent allergic reactions in human beings. About 70-some-odd%, probably more than that now, of the grains we consume have been genetically modified today. And that's important because we're able to double the outputs, or we're able to use less pesticides, we're able to do a series of things that increase the productivity. And it means we don't have to cultivate an area the size of the Amazon to currently produce the grains we have. To my knowledge, so far, and if anybody here has evidence to the contrary, please bring it up, there has yet to be a single person who's died eating a genetically modified food. That is an extraordinary record, okay? Because people have died eating mushrooms, and people have died eating peanuts, and people have died on this, that, and the other. That doesn't mean that this is guaranteed. That doesn't mean that there will not be a period where we make a mistake with a genetically modified food or with a vaccine or this, that, and the other, but on the whole, life on this planet has been getting better. It used to be that 15% of people, 15 to 40, died in violent accidents. Today, even with Iraq, even with Iran, even with Congo, it's 2%. It used to be that people lived 30 to 40 years. We're now living, if you're seventh day, an average of 87, 88 for the population as a whole. We now understand that there's a whole series of centenarian genes, and if you have these genes, there is about a 77% likelihood, unless if you're an alcoholic or have a particular risk, that you will live to be 100. And as we begin to understand this stuff, there is stuff to be afraid of, there is stuff to regulate, there is a role for government, there is a role for regulation, there's absolutely a role for ethical debates. But we also have to understand that this is the kind of thing that changes governments, changes countries, and changes religions. If your religion is everything revolves around the earth, and then it turns out, Or if your religion is if you don't sacrifice 100 virgins, the sun won't come up, and then the sun comes up, then there's a dissonance between reality and what you're preaching. And we're going to enter an era of rapid speciation, not just of industries and businesses and countries. We're also going to enter an era of speciation of a whole series of governments, religions, beliefs, because this stuff is going to challenge stuff in fundamental ways, okay? I mean, when your grandparents were around, there was exactly one way to get pregnant. There are now dozens. And your kids are going to deal and have questions that you aren't even beginning to dream about. This is a really interesting, complicated, fun period to be sitting, whether you're a social scientist or an ethicist or anything else. Let's take 3 or 4 more. Okay? Shoot.
I have a question adding on this. Our natural cells are billions years old and they modified according to nature and environment. But now we have a synthetic cell, and these cells, if they fuse or modify with the natural cell, we have no control. We can make any industry, which we can stop anytime, but once it is bio industries, bacteria are out or fusion is out, no way we can control. How the politics, scientists, and other people thinking, how we'll control if any bio disaster happens, that 2 cell fuse and something happened, which we don't know. Our experience is today in nature is billion-year-old.
Yeah, and you've got to really think about that. I mean, look, I think we have to call every portion of society into account. And I think NGOs have been particularly good at raising a whole series of questions. But I also think, just as there have been industries and companies that have made horrendous mistakes, there have been NGOs that have advocated things which, to my mind, are galactically stupid. One of the things that one of these NGOs advocated was when the first genetically modified seeds were put out, there was an instrument in there that said this seed will never reproduce. And they went on a global campaign and forced the company to take out what they called a terminator gene. That is really stupid, okay? Because one of the things that I would like to do with any type of a genetically modified organism is make sure it doesn't freely reproduce. Make sure it's a 1 generation or a 10 generation or 100 generation. The second thing you want to do is make sure it's biocontained before you understand for years what the effect of this thing is. The third thing is make sure that these things have a nutrient shutoff in such a way that if these nutrients aren't present in this particular combination, that thing is unlikely to grow. And the fourth thing is figure out something that will kill this before you release it. So put a whole series of controls into any of these things, because a whole bunch of stuff you'll do it right, but we will make mistakes. And we have to be ready to cope with those mistakes. And we have to, really be thinking in terms of safety of this stuff. And what drives me crazy is when people don't see the trade-offs in that stuff, and they make an argument like the poor farmer won't be able to harvest his seed. Okay, I get that. But when you're introducing stuff, yeah, you really do want terminator genes in this stuff. And everybody should be held accountable on all sides for this. There aren't good guys and bad guys in this stuff. There's a whole bunch of people who are trying to do the right thing according to how they see the world. And then there's a few people who we don't want to clone. Shoot.
The factors that you just brought up were very mature, very thoughtful preventions of anything.
Remind my wife.
Very mature. But given that you're discussing the fact that a lot of these come from startups near universities in two-story buildings, what kind of protections are there, and what kind of either regulations or medical ethics boards, et cetera, are there so that people are as thoughtful as you are? Because in the wrong hands, a lot of those things won't be successful and will be shunned, and people will become scared instead of excited.
You know, one of the people I really admire is Drew Endy, who's been working on this stuff. He's a wonderful professor. He used to be at MIT, then for some reason moved to the West Coast. And he claims it was for the brains. I think it was for the weather. But as you're thinking about what Drew has been building and creating, he's created this incredible organization called iGEM, which is the International Genetically Engineered Machine Competition. And some of you have heard Dean Kamen speak, the guy who built the Segway, and you know that he built this great robotics competition. What Drew and a bunch of people have been building is a jamboree every year where kids show each other what they built over the last year that's alive. And they show up with the most extraordinary stuff. They've built the equivalent of a Radio Shack for biology, which is open source, and you can go on the web and get a standard registry of biological parts. And you can get your sequences of DNA, and you can get your sequences of RNA, and you can get a cell-free system or an E. coli or a B. subtilis or whatever, and then you can make stuff. And these various college teams are showing up and making stuff. So 2 years ago, the team from Baylor showed up, and they had discovered, or somebody had discovered, that there's a stuff inside red wine, resveratrol, that's supposed to be good for you. And they engineered that into beer. Why that would occur to a college student? I have no idea. Well, this year they got a little bit more interesting and exotic. So the team from Tokyo showed up, and the team from Tokyo was trying to build a bacteria that could do 4 things. It was trying to build a bacteria that didn't get sunburned, that didn't freeze, that didn't react to violent changes in temperature, and that would eat iron and excrete oxygen. Well, that's a nice little bacteria. That's kind of interesting to have around. Why would you do that? Because if you stick this stuff inside a spaceship and shoot it at the polar ice caps of Mars, you will get a viable colony of bacteria that is creating an oxygen atmosphere around that planet. So these kids were trying to talk about terraforming a planet using this kind of engineering. These are not subtle projects. And as these technologies spread, there are really good questions as to how people are going to use them, where they're going to use them. And I think the good is going to far outweigh the bad. But we do have to keep track of certain gene sequences when they're ordered out. We don't want people assembling certain things. And there was a huge debate that took place not that many years ago, 5 or 6 years ago, as to whether the flu sequence for the 1918 flu should be published or not. Because one argument is if you publish it, somebody will eventually build it. The counter-argument to that is the Wikipedia argument, which is if you publish it, then you'll get 100 groups creating a vaccine before anybody comes close to being able to build that thing. Let's take 2 more. One in the back over there. And random. I heard your question, how do you get funded? A whole bunch of poor innocent foundations and universities and family offices come to us and tell us to go try and find creative ways of losing them money by building up companies, and we've been very lucky so far, and we've had some success in the companies we've built, and we go out and do it again. Well, the question is, how do you know this will happen in America? Because the first map of global nucleotide data flow was done by 3 of us. And what we did is we tried to figure out how much gene data is coming out of these machines, where's it moving, how's it flowing, and who's using it. And through a few years ago, about 54% of all the gene data in the world was being produced and consumed in the United States. There were a couple of pockets in Europe. Britain was large. Finland was there. France was there to some extent. Italy was there. Germany was there. Japan is a relatively big node. China was almost non-existent. So it's the U.S.'s game to lose. Shoot.
Yeah, my question is, what scenario could you paint that would, with the development of this type of technology, where the current notion of God is basically fundamentally brought to question as we currently know it?
I knew I should have picked this side of the room. Look, I mean, everybody's got different beliefs. I'm actually writing a book on that, what some of this stuff implies for societies and what it implies for religions and what it implies for industries and stuff. Let me give you, without trying to be facetious, 2 things to think about. I'm not going to give you a whole framework because that's a chapter. But if you can take a skin cell, de-differentiate it, and build an exact copy of your body, those religions that for years have been preaching resurrection and recycling and reincarnation suddenly become a little bit more relevant in terms of what the technology is catching up to. Right? And there's a whole series of places where you see it, right? Because you can now have a child that's born in 100 years. Because you can freeze sperm, you can freeze an egg, you can have a surrogate mother, you can freeze an embryo, and there's absolutely no reason why you can't have a child born in 50 years, and then another in 100, and then another in 150. But what this little experiment with the mouse does is it makes it feasible to copy a life form out of any cell. And then it starts to get interesting, right? I mean, our belief systems aren't quite there yet. There's some hints at it. Second way of looking at this is, you know, even for people who have a very strong ethical, moral, religious belief, I think one of the things science does is there's 4 big questions out there, right? There's who and why, and no scientist is going to answer that. That's the realm of belief and religion and stuff. And then there's what and how, and that's the realm of science. When religions try and explain the latter 2, they get into a hell of a lot of problems because they say, this is impossible. Then all of a sudden, you know, it turns out Copernicus shows up, or Galileo shows up, and game over. Or it turns out the god of maize isn't quite as productive as you thought he was. And then it's over. And I think as we begin to discover a universe that's a whole hell of a lot bigger and more complex than we think, if you believe in a higher power, that higher power becomes that much more powerful in the measure that you understand that what he created is so much greater than what the current local rabble-rouser is preaching. Last one. Where's the mic? You pick.
So nice of you. Can you demystify the whole cloning evil folks in terms of you have genetic identical twins where through mutations, you know, through their development, they don't really turn out to be alike. So potentially you would actually have identical copies of genes, but have a completely separate individual in terms of how they think and who they are. Because I think it's providing some sort of stigma towards the genetics industry. So we should try and maybe get rid of that. What do you think?
So look, if you were to clone yourself, you'd have an identical twin that was quite a bit younger. And it would be very weird because there would be about half the stuff that twin did that you'd say, my God, that's identical. And then there'd be about half the stuff that twin you'd kind of say, where did that come from? And you can see that when you have 2 children out of the same parents. Where did that one come from? Now, what's really interesting about this stuff is we're beginning to learn how complicated this stuff is, and we're also beginning to understand just how quickly evolution can take place. There's a really interesting article written or published by three groups simultaneously about 3 weeks ago, talking about adaptation of Tibetans versus Han Chinese to high altitudes, and how the Tibetan genome has a way of absorbing oxygen in a different way from lowland, and by the way, that has a huge impact on live births, among other things. So there's a big selection pressure out there. That evolution, the argument is happening across thousands of years. But what's really interesting about what's happening today is we're beginning to find that evolution occurs in real time. And one of the most interesting labs in the world is right here. Right? So the science that you founded of epigenomics is beginning to tell you that these adjustments take place inside cells, and that there are times when cells are particularly vulnerable. And if I get this wrong, please come up and correct me. So I'm quoting my teacher, among other things. But the argument that is being made is that there are times when it's almost like you're opening the door to change when you're getting implanted, as you're being born, as you're a young child, when you get elderly, where your genome is much more vulnerable to changes, and that we're doing most of our pharma studies on people who are healthy. And we're not doing those pharma studies or those chemical studies on people during the periods of life where their epigenome may change in a fundamental way. And if we were doing it right, then we'd be studying the impact of chemicals 30 years ago, not today. We'd be studying the impact of vaccines 30 years ago. We'd be thinking about medicine and how these changes take place and how they take place in real time, because that evolution can take place very quickly, and it can take place in real time. And there's actually evidence. There's a wonderful book called The Beak of the Finch about people who went back and studied Darwin's work in the Galapagos that tells you this stuff actually can happen really quickly. And epigenomics is, I think, along with metabolomics and other stuff, one of the most interesting single areas of research today. The other really interesting area of research today in life sciences is beginning to understand our organisms as very complex symbiotic beings. We probably have about 100 times more cells in our bodies that are non-human than are human. And those cells have evolved around time to allow us to breathe, to allow us to do digestion, to allow us to break down stuff, to allow us to not have stuff happen. And sometimes they go rogue and we come down with horrendous diseases. But for the most part, we're this very interesting symbiosis of organisms. And as we understand that, it's not just the human genome we're going to be interested in. We're going to be interested in this whole microbial genome that makes up all of our bodies. This is an absolutely incredible period of discovery. This is a period where we are going to find stuff that is just mind-boggling. It will bring ethical debates. It will bring ethical quandaries. It will make some countries rich and some countries very poor. But it's a really interesting, important time, and you have to go out and make your kids aware of life science literacy, just as you've made them aware of digital literacy as they run around with their video games and their iPods and their Google research and the rest of the stuff. It's just going to be a part of a continuing education process, and hopefully we can do it in such a way that's not boring. Thank you.